Serving on her thesis committee a while back. A great pleasure for sure, and great to see her here. And she's going to be talking about lessons from retrospective FEP on NAMPT. It lists her as being at Pfizer. I believe she did this work from Genentech, and now she's at Blueprint right down the street here. So,
thank you.
Looking forward to it. Yeah,
Thanks. Yes. So today, let me just see if this works real quick. Okay, it works. So today I'll talk about some of this work that we've done at Genentech. When I was a postdoc, towards the end of my postdoc, Genentech was interested in evaluating FEP, and there was some internal prospective evaluations going on, and Vingxing is sitting here. He was very much involved in that work, but they also wanted to evaluate on a retrospective project, and this has been a collaborative work with Jen Knight from Schrödinger. She's not here today, but we picked NAMPT as a target because, well, first of all, Genentech was not interested in that anymore. It was a program that was killed over 6 years ago back then. But they had a lot of compounds made and tested. We had IC50s for over 1,000 compounds and dozens of co-crystal structures internally. They were available, and across these crystal structures, the binding pocket seems remarkably similar, so there was not much motion, so we thought this would be a good system. And also, something I thought would be interesting, because NAMPT is an oncology target, resistance would be an issue. So there was a concerted effort at Genentech at the time trying to predict these resistance mutations from cell culture work. So I thought this would be a good case for us to test protein FEP as well. So just for those of you who are not familiar with NAMPT, it's nicotinamide phosphorodiol transferase. It catalyzes the rate-limiting step in salvage pathway, and that generates NAD, and many tumor cell types seem to be relying heavily on NAD. So because of that, that was an oncology target, but I think because of biology, the program was killed. So although we thought originally it could be a good system, when we started working on it, we quickly realized that there were some complications with the system. Well, first of all, NAMPT is a homodimer. It has two active sites, basically. And in these set of FEP calculations, we've done perturbations only in one site. So I haven't been able to find any experimental evidence in-house or published externally that reports any kind of crosstalk between the two ligand sites, but nevertheless, that's an assumption that, so basically we kept the crystallographic ligand intact there in these simulations. And another complication was for NAMPT to be active, to retain catalytic activity, it has to be phosphorylated on a binding site histidine residue, but none of the crystal structures, over 100 of them, actually described the active state, the phosphorylated state of the enzyme. But in some of those crystal structures, there were some phosphate ions from the crystallization buffer that was bound in some, well, to be precise, 3 separate sites. So that was a complication. And an added layer of complexity, some of these ligands, they had a nitrogen-containing heterocyclic group that extends towards the co-substrate binding pocket, and they form a covalent adduct with the co-substrate PRPP, so you can imagine that might impact the IC50s. But with all of that in mind, we set out to, well, also actually, let me just mention one more thing. We had IC50s for all these compounds, but we had only the KD by actual biophysical binding data for only a fraction of these compounds. And for that fraction, IC50 did not really trend well with KD. So if you calculate, and I think there's some reasons for that, but I think the point that I want to make here is if you just calculate a mean unsigned error of the experimental IC50s between the experimental KDs, that gives you roughly 1.1 kcal per mole. So that's basically the experimental resolution that we have. That's right there is setting a practical limit for FEP to be able to reproduce experimental data. We picked around roughly 140 unique ligands from 6 different series, and we put them in 10 different maps, most of the time overlapping maps. And we had over 425 perturbations. I think after the analysis, removing some bed edges, I think we ended up a little bit over 115 perturbations. And I'm going to show you the results, but I think, so I want to focus on some of the learnings that we gained over the course of this exploratory work in terms of setting up the best practices for FEP calculations, both the setup and the analysis. And some of them also, maybe I should mention that I didn't have any FEP background before. I have extensive molecular dynamics and molecular modeling experience, but I was completely new to FEP type of calculations. So what I have found over time, the protein and ligand preparation, they're extremely important, and I'm going to show you some examples of them. And also what you put in the map in terms of the system and how you generate the map as well. For example, this is what I was talking about, sort of protein preparation. So I think for me it paid off a little to dig into the literature a little bit and try to learn more about the biology of this very complicated enzyme. And turns out the phosphorylated histidine stabilizes the pocket, and it actually better captures the nicotinamide, the co-substrate. But then when I did MD simulations, and these are like 100 millisecond MD simulations, with no phosphorylation, no phosphates, the binding pocket was very floppy. When I did an in silico phosphorylated version of my protein, it was a little bit better. And then I decided so I played with those 3 sites a little bit, and I eventually decided to keep these 2 crystallographic phosphates, which normally, if they're not necessarily a cofactor, when you set up an MD simulation, you just delete the crystallographic buffer components. Like you don't want things like that, or glycerol, and so this was helpful to reduce the noise that we were getting, certainly from these calculations. So this was one of the very first maps that I set up, being a naive FEP user, just following the user manual, and I guess I don't have the average error here, but so I'm going to give you some examples. For example, I was following somewhat an automated workflow of ligand preparation using the default settings. And turns out this particular ligand, which was very centric to my map, was incorrectly, its protonation state was incorrectly identified. So keeping it there in the calculation as a connector in the map is not really a problem, but as soon as I assign the experimental value to that, at that point I was actually, it's equivalent of a mismeasurement, if you will. So that definitely impacted these delta G conversions. And another thing that I was not, I didn't realize, I was docking these compounds using a docking protocol, but then I quickly became aware of the fact that they had to be perfectly aligned. You can't just dock them and thinking that it's a good enough. And that can be a problem sometimes, especially if you don't have a good initial crystal structure. Like, where do you start from? What are you going to align your next set of compounds? And another one, this particular one, I mean, you can see it's a very ambitious perturbation, but that was not flagged. But then you can clearly see that that's an outlier. So going through, so this is actually a very nice way of, so we had assessing the relative damage that each of my mistakes into my map, basically. So if I remove that very large perturbation, I already dropped the, so basically there's 2 edges per that ligand, and the mean unsigned error associated with those 2 edges is already 10 kcal/mol. So when you just remove one ligand, your mean unsigned error drops to from 2 kcal/mol to 1.5. And on top of it, if you remove the edges that were connected to the ligand with incorrectly assigned protonation states, that seemed like that actually didn't make a huge impact. You went from 1.5 to 1.4, but seems like the alignment, the ligands with inconsistent ring alignments, if you remove those, then the error drops back down to 1 kcal/mol. And fixing all these, and also something I noticed is the default map that comes from the software is almost never enough. You have to go back in there. It provides a great starting point, but the map needs to have a little bit more edges, because in this particular very limited experience, it's we lose a lot of edges basically. So with that, I mean, with all these following best practices, you can see the correlation plot gets tightened a little bit. And in terms of the results, so here I'm showing you the accuracy over edges, the delta-delta averaged over delta-delta Gs, and accuracy in ligands, and we are, and most of the compounds are within 1 and 2 kcal/mol. We're around 1 kcal/mol, which is very close to what's published and what I'm hearing today as well. And if I looked at the individual maps, and this is just a table that shows for individual maps how many ligands, how many edges, the average FEP similarity, average molecular weight, rotatable bonds, the experimental affinity, and my errors. So, for example, this serines, the oxypyridines, they were the largest of my series, and they seem very flexible, but they have very low similarity, around 0.5, but they're very potent nanomolar compounds. And when I looked at some of those trajectories that I pulled, the compounds seemed very stable in those. So we ended up getting very stable, very accurate results despite the low similarity there. But these compounds, a little bit higher similarity, but they're smaller, they're weaker binders, there's a lot of mobility in the trajectories, the binding poses, so the accuracy is lower. And then for the mid-range, mid-sized compounds, again, the similarity is low, and we didn't get much accuracy there. So I guess overall, like I said, we're around 1 kcal/mol, but to me it was interesting to see that there's not really, I mean, high similarity is not going to guarantee high accuracy, or low similarities doesn't mean that you're not going to get accurate results. So it's really a combination of series-specific features that ultimately impacted the FEP accuracy. And this is just an example of the mobility of the smaller compounds that I was showing you, telling you about. So this is 2 monomers, nicotinamide. I should have colored this differently, but this is the nicotinamide crystallographic binding mode. And in the simulations, you can see nicotinamide is sampling all over the binding pocket. But despite this mobility for smaller compounds, there were a few cases where FEP was able to predict some activity cliffs, although I think they were over-predicted a little bit. So in retrospect, maybe now, if I go back and just analyze the delta-delta Gs from the first 1 nanosecond of data, maybe now we can get more realistic numbers there. But usually, if I go back to the trajectory, usually I can make sense of what kind of interactions are being lost with that small chemical modifications. And this is an example of sampling, inadequate sampling. So these are two equipotent compounds. Experimentally, they're similar, but FEP was over-predicting the potency of this ligand. But then this compound was coming with very high errors in a lot of different edges. So when we looked at it a little bit more carefully, and we decided to run a single edge FEP for the same compound, but starting from a different rotamer, so basically flipping the binding pose a little bit. And then you can see, actually, if you go from this to this, so it was the sampling here that was, but it was converging locally. So sometimes it helps throwing these alternate conformations in there. It doesn't mean that you have to do it for every single compound, because even if you do it once, there's going to be other things that this is going to look similar to in the map. Okay, so another example is this particular compound. This is a low energy conformation. It's QM optimized, and every computational chemist, when they look at this structure, they think this should be the binding pose. But in the crystal structure, this is the binding pose, and I generated an omic map, and it seems clear. Although there's still computational chemists that don't believe that structure. But when I run a single edge, so ideally, in the perfect world, the delta G should be 0 between the two. But I was getting, and any non-zero value should be due to the bias in the initial pose where we start, right? So, and over 5 nanoseconds, FEP does predict around 1.2 kcal/mole, more favorable for this one. And I think, to me, coming from a protein packing perspective, so it's just, it just packs better there. Okay, so if I still have time, I think I can mention just, I have a couple slides on the resistance. So as I mentioned, when this project was active, I think there was a concerted effort of trying to predict resistance mutations for this oncology target in the absence of clinical data. So basically at Genentech, they did they selected for resistance mutations by treating some cell types, cell cultures, with these compounds. And then they identified mutations that came from those that were picked up in those selection passages, basically. And there were some active site mutations, mostly glycine 217, and then there was a distal mutation, serine to tyrosine or phenylalanine. And so I did a very quick experiment with putting FEP. So basically, first I did an alanine scan in the binding pocket residues and see if I was going to pick up a signal at all. And I did pick up a signal at glycine, so that was very interesting. But then when I compared it, so, and also in my mind, I was thinking, whatever mutation that's coming up, ideally it shouldn't be affecting the substrate binding, but only the inhibitor binding, otherwise it wouldn't be picked up as a resistance mutation. So I used the nicotinamide as my control, and it looks like these kind of mutations at glycine 217, they seem to be preferentially impacting inhibitor binding, but not the substrate binding. So I thought this was interesting. And another experiment that I did, so this is the serine 165, which came from that resistance selection experiment in vitro. And I picked randomly equal distance sites. The only criteria I had was their site chain pointing into the protein and not necessarily exposed. But that was pretty much my only constraint. And then I did an alanine scan on those residues, and In this particular case, I didn't get a nice convergence, but I was consistently getting some convergence issues. And this actually, I think Jen and Anthony from Schrödinger, they're looking more into it. And turns out it's basically this larger side chain, or actually even in the smaller case, it's right against the helix, and it's having some trouble rearranging that helix when it's bound to the inhibitor. But with the nicotinamide, there's more room there because nicotinamide is smaller. So this is certainly not giving us the mechanism of resistance just by doing this quick scan. But I think if you think about it in the context of a team of scientists doing cell culture assays, biochemical characterization, and there's crystal structures as well. So I think having this sort of a flag there, I mean, it's really not a big deal to go back in the lab and then do one site-directed mutagenesis and test this. So I think in retrospect, this would have been very interesting to point out. So I think this is really what I wanted to tell you today. Like I said, in my experience, it pays off to invest a little bit time in properly setting up, sort of like getting more familiar with the protein system, enzyme system, the biology of it. And then sort of following these best practices. It's certainly not a button to push FEP that you get these magical numbers. And I think the protein FEP, in my experience, is very promising for tackling questions like drug resistance and selectivity, I think. And yes, and I think overall, in this part, just to summarize in one sentence, I think for the ligand series for NAMPT, I think FEP was able to separate weak and tight binders, but I don't think it did a great job in rank ordering within series, which I guess is obvious to this crowd, but it was not to us when we started working on it. So thank you very much.
Yeah, over there.
So your data set, there were over 400 mutations, right? 430 or so, I think, in your
Perturbations, yes.
edges.
Yes.
And I was wondering how many of those actually alter the net charge or the ligand or the system?
No, so we did have some charged compounds, but we had to, so we were told actually at the time that that it wasn't basically implemented in the package that we were using. So we actually removed those charged perturbations.
It's counted in 2 ways.
Did you try, for your torsional sampling, did you try using custom core, because that would include that area to the rest sampling, and it should?
So Jen actually tried that, and in some cases it worked, and in some cases it didn't, but we didn't really do a systematic analysis and sort of figured out which cases that it worked better.
You have in front?
Yeah, hey, in the cases where you needed sort of the accurate ligand alignment to get good results, did you ever try just brute forcing a very long FEP to see if it converges to the same result just by having a lot of sampling?
Yeah. So I don't know if you would agree that it would be brute force, but so we did extend it up to 20 nanoseconds, but that was not enough. But I don't know if it would make a difference. But I also tried increasing the lambda window a little bit. So that helped with, I think, more ambitious perturbations than the sampling issue. So yeah, basically to answer your question, 20 nanosecond was not enough.
Yeah, a quick question about you had fragments and you said that they were kind of weak affinity and you showed that they move too much. Is that related just to the fact that they're not big enough to bind in that site optimally, or is it just that the interactions are not strong enough? So can you deconvolute those two things? Do you have some potent small molecules that you could also compare with to see if they do stay in place?
I don't think we had potent small molecules to make that distinction, but NAMPT does have a big binding pocket. And even maybe so I guess the underlying assumption was because there's a crystal structure of that fragment, that has to be the binding pose, but that may not necessarily be true. So I get, we didn't have those compounds to dissect.
All right, great. If there's no more questions, let's thank Ayşegül again.
Thank you.